amplified by the RNA guide strand. If you do a double-stranded break, that's unstable in the cells, and it's rapidly healed by a process called non-homologous end joining. And this will join the two ends together. If during this process, there's often base trimming such that the repair is imprecise and you end up damaging the gene. In fact, you make frame shifts and you can destroy gene function. If you end up making a perfect repair, well, then it remains to be a target for the CRISPR enzyme, and it serially will cut until you end up with something that can't be cleaved again. And so this destructive process can be extremely efficient, and it's not difficult to get 90 to even 99% efficiency of gene destruction. If you want to build gene function, you can add additional DNA elements in during this process, which can be short oligos or long fragments that contain far more information. And a short oligo could be 100 bases, 200 bases long, that contains homology arms to the area flanking the double-stranded break and whatever new information you're encoding. And that could be as small as a single SNP corrections. The long double-stranded fragments can add kilobases of new material, but in all of these cases, you have a homology arm to direct where this happens. Unfortunately, if you just add these reagents together, you typically get 1 to 10% HDR efficiencies, and you've got to take steps to improve HDR. So what can you do to improve HDR? So the first step in improving HDR is to work with the best possible guide site. And different sites may cut seemingly efficient, but they actually, in the short time span of doing HDR, different sites are more active. And so you need to test different sites. Another is that especially if you're using short single-stranded material, all of these synthetic DNAs are rapidly degraded. So you need to chemically modify these to allow them to stay in the cell longer so that HDR can actually occur. And then if you're using short strands, strand selection can actually be important. Another factor is the fact that you can have recutting. And if you're making a small number of changes, you can still remain a substrate for the CRISPR enzyme, and you'll get recutting such that you get HDR and NHEJ and don't end up with a perfect edit. Finally, if you go to longer insertions with HDR, those will obviously all no longer be a substrate for the CRISPR enzyme since the guide site has been radically changed. But then you have to deal with blunt insertions. And finally, you can use compounds that enhance HDR through some different mechanisms. And we will consider all of these in the ensuing talk. So some of these will depend too on what format of HDR template you're adding. If you're making small changes, you will use short single-stranded oligos, which are typically 100 to 200 bases long. You can use long single-stranded DNA for bigger edits, but while we originally, several years ago, promoted this, we found since that there are just too many internal deletions that occur with this when you're working with somatic cells. For oocyte injection, this is fine, but for somatic cell work, it's not. So now we're working mostly with long double-stranded DNA, which can be linear, which needs to be chemically modified, or circular is plasmids or even better than nanoplasmids. We're also investigating long circular single-stranded DNA, but I won't talk about that today. Now, if you're using a short single-stranded DNA, an oligo, as an HDR donor, the distance of your edit from the cut site is critical. So this is a plot showing the cut site at 0, and then the efficiency of inserting a 6-base EcoR1 site as you move to the left or right. And as you can see, there's a very small window around the cut site that this is efficient. So if you're doing this, you need to have your cut site and your critical mutation be near each other. Also, when you're using single-stranded DNA, which strand you use can matter. So if you're close to the cut site, either the targeting or the non-targeting strand will work fine. But as you move further away from the cut site in the PAM distal direction, using the non-targeting strand works much better. And conversely, if you're heading in the direction of the PAM, the targeting strand works better. So it's important to recognize which side of the cut site you're working with, which helps determine which strand you make for your oligo for HDR. Now, if you're using double-stranded DNA, obviously this is irrelevant because both strands are present. The other is, how do you chemically modify these? So this is a plot of examining a whole host of different chemical modifications, with the bar at 1 being the efficiency of unmodified DNA. And if you look at a lot of different modifications, strangely enough, even some reduce HDR. But I'd use as a start point the addition of some phosphorothioate linkages. Just 2 phosphorothioate linkages on each end will significantly increase your HDR efficiency. But you can add additional modifications beyond that. And we have found a number of different modification patterns that substantially improve HDR, which are just standard, simple things that you can add just by clicking that on the web ordering site. Now, the other area of a real interest when you're doing these small edits is recutting. And recutting occurs when your change is so small that the CRISPR enzyme still recognizes that site as a target. So what we studied here was looking at 2 different sites and 2 different cell lines, making every possible base change, walking to the left and right of the cut site, looking to see what changes would actually improve getting the final outcome to be the perfect HDR edit. So this is introducing silent mutations, things that change the DNA sequence and therefore change the reactivity of the CRISPR enzyme with guide to this target, but don't change the sequence of the outcoming protein. And we've mapped what changes have an impact on guide cutting and therefore are worth making. And this plot here is a summary of all of these experiments, which shows on the left the red bar showing that this edit was, depending upon the setting, anywhere from 1 to 5% efficiency, which is kind of terrible. And if you introduce these silent mutations, you can increase that to about 4% to even as high as 35%, just by this addition of silent mutations. And so you can add chemical modification, choice of strand, silent base changes to dramatically improve your editing efficiency when using short oligos as your HDR template. And you don't have to remember all these details now. All of this was published a year and a half ago in scientific reports. Now, what about larger inserts? And we are now recommending use of double-stranded blunt DNAs, or you could also use circular DNAs, and in which case we recommend the nanoplasmids from Aldevron. But if you're using a blunt linear DNA, you can either get the desired HDR, where the homology arms undergo homologous recombination and you get a perfect insertion, or you can insert blunt, in which case, if you're at the desired edit site, you duplicate homology arms, or you can even insert blunt in the irrelevant sites, because a blunt piece of DNA can insert in any double-stranded break at any location. So how can you reduce that? Well, and one of the ways is to put chemical modifications at the ends of the linear double-stranded DNA. These can reduce exonuclease attack, and they can prevent blunt ligation. So this is an examination of a whole series of different mod patterns, where the unmodified starts as a base equals 1, where the gray bar is the HDR efficiency, the light blue bar is the blunt insertion efficiency, and then the dark blue bar is the ratio of HDR to blunt. And you can see that as we vary the different modification patterns, we can reach a pattern where you can have, in this case, it's a kilobase size, it's a 42 base insert with 200 base homology arms. You can increase the HDR rates, decrease the blunt rate, and dramatically improve the ratio. Now, that's not the only way to improve this HDR efficiency. Another way is to use compounds that affect the cellular machinery. And there are 2 competing pathways for healing this double-stranded break. One is non-homologous end joining, which we've talked about, that leads to the creation of these imperfect joinings and gene destruction. And you can get small molecule drugs that interact with the enzymology of this pathway and inhibit NHEJ. If you inhibit NHEJ, you shunt more of the repair down the HDR pathway. Well, we originally found one compound that was fairly active, but we've now got a better compound, which we call our version 2 of the HDR enhancer. And it's a NHEJ inhibitor that works with reasonably high efficiency and relatively low toxicity. And so that's an easy, inexpensive way to increase your HDR rates. Now, in some settings, though, this may be undesirable because inhibiting NHEJ can also lead to other kind of chromosomal events and increase inversions and translocations and large changes. So if you're basically just creating cell models and need efficient HDR, this is a really fast and cheap approach to do it, which is very effective. Now, what if you combine these? So on the left, I have a study where we're looking at 3 different guide sites, insertion of a kilobase size GFP tag. And we're looking at these as the first 2 bars are long single-stranded DNA of either the targeting or non-targeting strand, an unmodified long double-stranded DNA, or a modified long double-stranded DNA. And you can see that without any enhancer treatments, that the modified double-stranded DNA works the best in all cases, but the HDR rates are poor. You're looking at best at maybe 15%. Now, if you add this NHEJ inhibitor or enhancer V2, you can see that you get a slight boost of the single-stranded templates, but the double-stranded templates now dramatically increase, and you can achieve HDR efficiencies approaching 70%. The other thing that we observed in this experiment was that in addition to doing Illumina short read sequencing through these to look at the junctions, we did Oxford Nanopore long read sequencing. And we found that many of the HDR events using the long single-stranded DNA contained large internal deletions. And so this is why we are no longer recommending long single-stranded DNA as a template for HDR. We're recommending long double-stranded DNA, which in these experiments did not produce any events that had internal deletions. What about the blunt problem, though? So if you look at an experiment where you're assessing the relative efficiency of blunt integration, which, as you recall, is undesired, you can see that these long unmodified double-stranded DNAs can have blunt insertion as high as 25%. If you use the chemical modifications, you can drop that to maybe 7 or 8%. But if you combine that with an NHEJ inhibitor, you dramatically drop those rates, because remember, blunt insertion occurs via NHEJ. And so this is now reducing NHEJ and promoting HDR. And you can see that even the unmodified templates are running at 5 or 6%, whereas the modified templates are now inserting blunt at around 1%. So now you're getting down to a level that's low enough to be usable. But what about doing HDR where you're not inhibiting NHEJ? All of these NHEJ inhibitors will have potential side effects of increasing inversions and translocations and other gross chromosomal events. The cell does not like to have double-stranded breaks, and fast repair is healthier. Well, it's a lot easier to inhibit a pathway than it is to stimulate a pathway. But fortunately for HDR, there is actually an approach to this. And that's a protein called 53BP1. 53BP1 is a natural inhibitor of HDR that promotes NHEJ. If you inhibit 53BP1, you could release this inhibition and have more active HDR. And indeed, that's been well described now. So 53BPM promotes NHEJ and inhibits HDR. And in January of 18, a group published that a small peptide of 74 amino acids could be created that actually ended up inhibiting 53BP1. Now, 53BP1 is ubiquitin binding, and this inhibitor is a mutated version of that binding handle out of ubiquitin. And so if you add that, you should be able to increase HDR without inhibiting NHEJ. So we tried that. We were very excited with this. But when we applied this in the setting of using CRISPR ribonuclear protein complexes that are transiently expressed and very specific, we didn't find that this inhibitor worked at all to our benefit. Well, maybe a little at very high dose. And the reason for that was the paper used this peptide using a sustained plasmid overexpression system. And this sustained super overexpression just isn't how you would do it for critical editing. You're going to be using short transient expression of the CRISPR components where you get a shorter duration of action and more specific editing. So we reasoned that if you took the same approach but evolved the peptide to be higher affinity, that maybe it would actually be useful. So we created a screening process wherein we took the 74 amino acid peptide and made every possible amino acid substitution at every base. And so it's a library of 19 times 74, which we put into a plasmid system and used it to regulate expression of an HDR event that was detectable in cell sorting. And we're able to sort mutants that improved HDR as opposed to left it the same or didn't. And if you did this several cycles of it, you could enrich for a collection of mutations that improved HDR. If you look at those, any single mutation at best maybe increased binding affinity by twofold. And that's really not that significant. But if you stack these mutations into 5 or even 10 of the mutations into the 74 residue peptide, you can increase binding affinity by as much as 100-fold. Now, when we take this mutated ubiquitin sequence and apply it into an HDR system, you can see now that these are having much improved HDR rates over baseline. And in fact, that it works at low dose, and this plot doesn't show it, but this high level of HDR improvement extends down to 1 micromolar, which is a level of use of the peptide that is compatible with RNP methods for doing CRISPR. Now, it's not just with these synthetic linear templates. We can also use this to improve HDR if you're using an AAV template. AAV has commonly been used as an HDR template, but when you're working in a clinical setting, getting GMP AAV is both expensive and time-consuming, and has certain toxicities if you increase the AAV dosage high enough to get high HDR. What we found was that you could use this peptide to dramatically increase the efficiency of AAV-driven HDR a many-fold, or if you want to just leave it at the same level of efficiency, it allowed for getting to that efficiency at about a 4-fold lower dose of AAV. Now, the question remains, did we really succeed in creating something that improved HDR but did not inhibit NHEJ? And in this experiment, what we see is an HDR template. So it's a short oligo, single-stranded, 40 base pair homology arms with a 6 base internal ECAR on one site. So this only inserts via HDR. So with putting it into 2 different sites, you can see in this case, you've got a little close to a 40% HDR efficiency that then boosts to almost 70% with this peptide, or even 80% with the NHEJ inhibitor. And similar boosts are seen at a different site. Now, if you go to a model of NHEJ, we are now inserting into a cut site a blunt 60 base tag. This has no homology arms, and so this cannot enter via HDR. It can only enter via NHEJ. So if we use no compound, you're getting about a 40% introduction of the tag into the cut site. With the HDR enhancer peptide, you see about the same result. Whereas if you use the small molecule NHEJ inhibitor, you almost completely eliminate the blunt tag insertion. So what it appears clear to us now, what we've done is created a compound that can be used to improve HDR without inhibiting NHEJ. So where's the value of that? This is what I consider to be sort of a poor man's survey of translocations. If we create a library of amplicons NGS sites that look at off-target effects, what you'll get is a forward 1 and a forward 2 that look at one site, a forward 1, a reverse 1, a forward 2, a reverse 2, a forward 3 and a reverse 3, and so on. And everything between a forward 3 and a reverse 3 is that site. If you get any NGS sequence that's between, say, a forward 1 and a reverse 3, well, that's linking two things into an NGS read that don't exist next to each other in a normal genome. And it's evidence for the existence of a translocation. So if we look at these libraries of amplicon NGS sites, with no enhancer, you get a very low translocation detection rate. With our HDR enhancer peptide, it's identical, very low. It's basically a background. But if you use the small molecule HDR enhancer, which is an NHEJ inhibitor, what you see now is readily detectable translocation events. And therefore, what we've done is clearly created a HDR enhancer compound that does not inhibit NHEJ and therefore does not come with the added undesired side effects of increasing things like translocations and inversions. So in summary, using CRISPR, it's very easy to destroy a gene. It's much harder to add new content. And the new content through HDR can be added by multiple methods. You can do that with single-stranded DNA oligos, with long double-stranded DNAs, with plasmids, with AAV. But all of these things need some kind of a treatment to increase their efficiency because the native efficiency is unacceptably low. And you can use strand selection. You can add silent mutations to prevent recutting. You can use chemical modifications to slow nuclease degradation of the template or to block blunt insertion. And you can use compounds which influence the cell's biochemistry for NHEJ or HDR. And that includes small molecule compounds that inhibit NHEJ, or this new peptide compound, which enhances HDR. So thanks for your attention, and I'll take any questions now.
Any questions from Mark? It's not too long. Oh, sorry.
I have a question, but I'm online, so.
Yes, please go ahead and ask it.
Hey, Mark, this peptide is pretty fascinating. Do you think it's possible, I guess I'm assuming it doesn't have any special modifications, that you could try fusing it to Cas9 or even the guide RNA, just kind of for fun to see if it can locally do that job, or if it has to be delivered separately?
What I'm showing you right now is just stuff. These last two slides are just hot off the press. And so we've got a peptide now. We have a sequence that's optimized that works very well. And now we're beginning to explore more how you exploit it the best. So this isn't available. This isn't yet a product. This is stuff we're still optimizing. Everything prior to the peptide is stuff that can be obtained easily. This is still in its experimental stage, and we're working right now with collaborators to beta test it and work out these details. Like, is there better ways to deliver it, or how is it best delivered? And there's all kinds of different things we can try now that we haven't.
Oh.
That was really fascinating presentation. You mentioned, I think, that you could make chemical modifications to the donor template to improve efficiency of HDR. And I think you mentioned one example was phosphothioate, but you said, I think, there were better ones. What would you recommend as the best modifications to make?
Well, what we're using as our preferred combination is phosphothioates plus a non-nucleotide N modifier on both ends. The exact nature of that we're not making public at this point, but they can be readily ordered through IDT.
So you can buy it, but you can't disclose what it is. It's not in the paper that you,
No, this is different work, and that's not in the paper.
okay, thank you.
Very interesting talk. Perhaps this is a naive question, but can you also help the cell to use more homologous recombination by simply synchronizing it in S phase? Would that promote more homologous recombination just because that's where the template DNA should be doing?
Yes. Yes, you can do cell cycle synchronization and do your CRISPR editing at the appropriate times to leverage S phase, and that will increase your HDR, but it requires other manipulations, which you can do in vitro, but you may not be able to do in vivo. So you're 100% correct.
Thank you, Mark. Let's thank Mark one more time.
Thanks, everybody, and I'm sorry I couldn't have been there in person.